Hi, my name is Emily Doherty, and I'll be presenting our project titled Towards Brain Metrics for Improving Multi-Agent Adaptive Human-Robot Collaboration, a Preliminary Study. This project was made in collaboration between the University of Massachusetts Lowell and Worcester Polytechnic Institute. Human-robot collaboration is becoming more prevalent in many workplaces. Applications that could benefit from this include workers in high stress and higher mental workload situations, such as UAV operations in the military and surgical teams in the field of medicine. Actions that robots could take to reduce the cognitive workload of their human partners are to take over complex tasks for the humans or to reduce the amount of information it is providing for a brief period. Something we do know more about is how humans collaborate in the absence of a robot, and how their brains often synchronize when working collaboratively and cooperatively with one another. We aim to study how introducing a robot partner in a collaborative situation can alleviate mental workload and its effect on human neural synchrony that occurs in collaborative teams. Our research questions asked if physiological data, including brain signals, could show insights to improve team performance, especially with a robot partner. And we also asked, can we use these signals to detect excessive mental workload during collaboration? This was a preliminary study in which 6 dyads worked collaboratively. We used functional near-infrared spectroscopy to study their hemodynamic signals in their brain. Specifically, we used a NearScout 16 by 16 apparatus, and this was a hyperscanning study because we were simultaneously recording the data of 2 people collaborating together. For our optode montage, we specifically focused on the prefrontal cortex and the superior temporal sulcus regions of the brain, as these have been previously implicated in neural synchrony, collaboration, and short-term memory research. We created a collaborative task that involved teamwork with other humans, a robot, or both. In the task, participants played a matching game, either by themselves or in a team, in which they first studied an image of 16 blocks similar to this one on the top right. They studied this image for 15 seconds, and then they matched the pairs of the face-down blocks by the symbol appearing on the block. There were 4 conditions: human-human, human-robot, and human-watcher, human-human-robot, and human parallel play. The human-human condition was collaborative, and the participants took turns picking up blocks to match up the pairs. The participants had to agree on which block to pick up before actually physically picking it up. Similarly, the human-human-robot condition was a collaborative team with 2 humans and a robot working together. In the human parallel play condition, the 2 humans participants played individual games beside each other. This served as a baseline condition similar to previous work. During the human-robot condition, 1 human collaborated with a robot while the other human watched the game, so that we could study any synchrony of FNIR signals between the 2 humans. The playing time was unrestricted to best mimic a real-life scenario, and our robot arm was remotely operated by a remote researcher who had video footage and was controlling the robot from another room. So here are our FNIRS results for the 6 dyads. We used NIRS Lab, which is a NearX provided software to run the generalized linear model on our beta value on our FNIRS data. We then performed t-tests to see how well the brain data was to what we expected it to be. So we did comparisons between conditions. So here you first see human-human-robot versus human-robot. And then it goes on human versus human-human, human versus human-robot, human-human versus human-watch, and human-watch versus human-robot. Firstly, we see that the human-human-robot condition elicited greater workload in the prefrontal cortex than the human-robot condition. The human alone condition has greater mental workload when compared to both human-human and human-robot conditions, in the temporal regions mostly. And then finally, the human watch condition elicited greater mental workload in the prefrontal cortex than both the human-human and human-robot conditions. For our study of the neural synchrony, we performed a wavelet coherence to detect the synchrony between the pairs. Significant differences between human-human-robot and human-human conditions were in the right prefrontal cortex, whereas the human-human-robot elicited less synchrony than the human-human condition. However, there was still more synchrony in the human-human-robot condition than in the human condition in the right prefrontal cortex. This shows that while a robot may affect the amount of synchrony a team of humans have with each other, they are still experiencing synchrony. The human watch condition had higher synchrony in the right prefrontal cortex than the human-human-robot, showing that there might be an impact on synchrony by how many humans in the team are actually working with the robot. While our results revealed that humans do respond differently when working with another human versus a robot, our results also showed that less mental workload is required when working with either of these types of partners rather than working alone. This is promising for the use of robots in many workplaces, as they could alleviate some cognitive load of workers without imposing more load than another human counterpart. Our synchrony results showed that humans still have neural synchrony when a robot partner is introduced, which is often vital to cooperative and collaborative communicative collaboration. Interestingly, our results also showed that watching rather than participating has greater effects on prefrontal cortex activation. We encourage researchers to explore this further. And for future works, because this was a preliminary study with only 6 dyads, we of course encourage researchers to recruit more participants with less robot experience. And FNIRS literature is now showing that the combination of EEG and FNIRS is very promising for a better view of the brain for both spatial and temporal resolutions and a more efficient measurement of workload. And finally, this research was supported by a collaborative seed fund grant funded by the University of Massachusetts and Worcester Polytechnic Institute. Thank you.